Hello everyone, welcome to this online lecture on medium pressure liquid chromatography. You are all very much aware that this lecture is the part of online course Advances in Chromatography. So once again, we are all together under the organization of DIPAM Foundation and Institute partner Sheth M Science College Patan for this online course Advances in Chromatography. Friends, in this Advances in Chromatography online course, we have particularly designed 12 to 14 lectures wherein we are going to involve few of the techniques that were left in the basics chromatography course along with the hybrid techniques that involve both chromatography and spectroscopy along with some of the mathematical and research based aspects that are involved in high throughput research in chromatography. So let us now begin with medium pressure liquid chromatography. Medium pressure liquid chromatography. Why we require medium pressure liquid chromatography? Earlier, before medium pressure liquid chromatography came into existence, we already had LPLC, that is low pressure liquid chromatography. It was one of a popular preparative column chromatography method before 1970s. So friends, over here we will focus along with this MPLC on a particular term that is preparative column chromatography. Just after this slide, we will be discussing what is preparative column chromatography along with its subordinate that is analytical column chromatography. The limitations of LPLC is the low sample loading, that is the quantity of sample that can be analyzed is quite low. But in 1970, overcoming of the limitation took place in LPLC and large sample loading was possible, which resulted into quick and efficient purification of a sample. Choice in stationary phase and reusable property of this stationary phase came into existence, and the operation became more simple and the cost of maintenance was decreased. These were some of the factors that could help overcome the technique of LPLC, and today we have MPLC. Nevertheless, scientists today having HPLC and UPLC as some of the most modern chromatography techniques. But when it comes to preparative column chromatography, MPLC and HPLC go parallel hand in hand. Attractive method for preparative chromatography is the medium pressure liquid chromatography. It is used individually or along with other methods of preparative chromatography. So this was just an outline about medium pressure liquid chromatography. What are the major advantages of this MPLC? Pressure is optimal. When we talk of low pressure, that is LPLC, pressure is quite low. When we talk of HPLC or when we consider UPLC, the pressure is too much high. And these kind of high pressures are very much useful when you want to do the analytical part of research. But when you are interested in doing the preparative part of research, you always require a medium pressure. And that optimum pressure is very much inherent or required for better resolution, which is adjusted through fine packing material. We very well know that as the size of the packing material, that is the stationary phase, goes on decreasing, the pressure goes on increasing. So the packing material size plays a very important role when it comes to MPLC. It is automated and an unattended operation, that is the minimum human interference is required when you have MPLC. Multi-dimension protein purification and purification of tagged recombinant protein can be done using MPLC. High resolution ion exchange and high resolution size exclusion chromatography are the very best applications of MPLC. So these are a few advantages that make MPLC even today a very good method for application. Now, let us understand what is analytical as well as preparative chromatography. See, be very clear. The thing that matters is not the particle size, but it is the objective of separation. If you go back to one of my initial lectures of basics in chromatography that even we had as a pre-course lecture in this series, I have very clearly mentioned that on the basis of the objective of separation, there are two objectives. One is a preparative separation, one is the analytical separation. Over here, I am just going to justify today that what is analytical chromatography and what is preparative chromatography. And be very clear, two more lectures that are going to follow in the next two videos, that is preparative HPLC and UPLC, will make very much clarity on what preparative chromatography is. See, be very clear that whenever you have analytical chromatography or analytical method, it is just for a qualitative as well as a quantitative determination of an analyte, and it requires a very small sample size, and the sample eluted is just disposed, thrown away, waste. But when it comes to preparative chromatography, separate and purify the component for further use. Means I have a mixture of components, I am separating out a component and I want to use it for further research. In such a case, I must have that compound which is separated in a good quantity, and when I need it in a good quantity at the initial phase before starting the experiment, I must also add it in a good quantity. So separation is possible, even purification is possible, but the component that is to be separated and purified for further use must be in a large amount, and therefore the analyte, the mixture, must also be in a large amount, and the sample eluted is not to be thrown away, it is to be kept for further use. Now, these methods of preparative chromatography, which are generally used to separate in larger quantity, to purify in the larger quantity, are good. But when you compare it with our traditional methods of precipitation, distillations, and things like that, at some instance you might feel that they are more costly. Yes, definitely they are. But industry today, hardcore researchers today, do not only think of cost effectiveness can be brought into science at a later stage, but they think of more purity, more accuracy, more preciseness. And when this accuracy, precision, and purity comes into existence, definitely preparative chromatography holds a better position compared to the traditional large scale purification methods. So, whenever we are talking of purification, always prefer preparative chromatography over other methods. Let us now do one thing. I have an animation of a preparative chromatography which I have got from NovaSep. And this NovaSep, where SEP is written, it stands for separation. And this separation somewhat is related to a SEP box that most of the natural product chemists or botanists use today in research. So let us just go through the video. This video will give us a very clear idea that what actually preparative chromatography principle has to do with. So let us have a look. I'll be keeping on explaining you along with the video. So keep your eyes on the video. You can see over here that we are mixing two components, a red color and a blue color component, and it is now fitted into this column and it is allowed to run. The mobile phase along with the components is allowed to run in a stationary phased column. You can see over here the two components are slowly and gradually getting separated. But see the quantity, it's a very large quantity. When the quantity is too large, generally we prefer it to be preparative chromatography. You can see the first component getting separated, and now you can see the second component getting separated. So this is where you need separation of the components from a matrix into a really large quantity that can be used for further experimentation, that can be used for further examination. You always will have a requirement of preparative chromatography, and this is a basic principle on an animated video form to make the concept clear to you. We are equally thankful to NovaSep for providing us such videos on Google. Now, let us move ahead. The medium pressure liquid chromatography. It is a broad spectrum of application in preparative chromatography. What are those? Let it be preparative MPLC or preparative HPLC. It is used for isolation and purification of molecules. If you go back to the video and consider that a red and a blue color sample were mixed and we got them separated, so it is isolated. Along with this isolation, we even got them in the pure form. So this phenomena is isolation and purification. But depending on the field of application, the amount of compound to be isolated or purified is different. This is where preparative chromatography comes into existence. If at all, I just had the requirement of 1 microgram of a compound or 4 to 5 microgram of a compound, analytical chromatography is the best. But why we will need preparative chromatography? If you will go through these three statements, you will be very much clear that why is the need of preparative chromatography. See, in life sciences, few micrograms of enzyme is more than enough for preliminary studies. But identification and structural elucidation of an unknown compound in chemistry or natural product chemistry requires few milligrams. When this word few milligrams comes, sometimes analytical chromatography cannot withstand this quantity, and in such a case you require preparative chromatography. Still moving a step further, isolation of standards and pharmacological testing demand larger quantities in grams. And this is the point where you want to isolate, separate, purify a component from a mixture in a large quantity, from few grams to 100 grams, 50 grams, 40 grams, 30 grams. Friends, preparative chromatography is must. And yes, medium pressure liquid chromatography, although works on a very simple principle like HPLC, but is having a fundamental base of preparative chromatography. Now, the instrumentation part of medium pressure liquid chromatography. Again, it's the same kind of instrumentation. You have a mobile reservoir tank, you have mobile phase delivering pumps, you have a sample injector, you have back stationary column, you have a fractional collector. This is a new thing, and then you have a detector. Pumps are also there, high pressure pumps. But as we have already described a lot about these high pressure pumps earlier, we will not much be focusing on high pressure pumps over here. In this particular video of MPLC, we will be focusing on three aspects, that is packed stationary phase, that is preparative column, fractional collector, and detector. Mobile phase reservoir is generally made of glass vessel and is a sonicator based vessel, so that through sonication the not required gaseous molecules will be thrown out of the mobile phase and mobile phase will be pure, which will help longevate the lifespan of the stationary column. Mobile phase delivering pump, these are reciprocating pump and have pulse controller to maintain the flow rate of the mobile phase. Consistent flow rate and accurate pressure can be generated through these mobile phase delivering pumps. 5 to 200 ml per minute is the flow rate of the mobile phase that can be maintained using these pumps, which have a medium pressure of somewhat around 40 bar. This is the point where you can make a differentiation between MPLC and HPLC. See how? If you think of HPLC, pressure in the HPLC can reach up to 200 bars. And when the pressure is too much high, we obviously call it high pressure liquid chromatography. But when this pressure is medium, 40 bar, let us keep the word medium pressure liquid chromatography. Moving still a step ahead, that is sample injector. The sample quantity is the most affecting factor here. The amount and volume of sample is very much important. Optimize the sample concentration. We will have a separate slide, separate explanation on what is this optimized sample concentration. Just wait for few seconds and we will be jumping on that slide. And this stationary phase, fraction collector and detector will be explained to you in details. Medium pressure liquid chromatography, this is the whole system of MPLC, where you have this A and B as the solvent reservoir. This particular part is this pumping and the injecting system. This particular is the samples that will be injected over here. This is the column where the separation takes place. It goes into the detector, and along with the detector it goes into the fractional collector, and the fractions are collected. For example, from this green sample, if at all there are three components, then component 1, component 2 and component 3 are automatically separated depending upon their lambda max from the UV visibility. Over here you will have a question that, sir, if I mix A, B, C and D and I want to separate them using MPLC, how would my machine separately set them in different collectors? That is the property of fractional collector. Your fractional collector is very nearly associated with UV visible detector. You can see over here, when your sample comes out of this column, it goes into the detector. In the detector, it is detected and it goes into a fractional collector. Let us assume your first sample to have a lambda max of 254, second is 454, and third one is 654. So until and unless your detector will detect the sample of 254 lambda max value, it will get collected in this first vessel. When the detector will shift from 254 to 454 lambda max value, the fractional collector will shift from first to second. And when it will detect from 454 to 654, it will shift to third. So friends, this is how separation using fractional collector is possible in MPLC. And this separate collection in a huge quantity make medium pressure liquid chromatography a preparative chromatography method. Medium pressure liquid chromatography instrumentation, which involves injector. The quantity and concentration of a sample in injector plays a key role. If you use dilute sample, you have to use it in very high quantity. So what will happen? When I am using a highly dilute sample in a very large quantity, injection of large sample is required, resulting into reduced separation efficiency. Because as the sample is highly dilute, the sample will be less and the solvent will be more. So separation efficiency may not be the required one. On the same side, when you are using a highly concentrated sample, then your solvent or mobile phase will be least and your sample will be too much more, which will lead to the tailing of a chromatographic peak, resulting into improper chromatogram. I have already discussed in the basics of chromatography series what is fronting and what is tailing. The graph, which is very perfect, when bulges from the start it is fronting, when bulges while coming down it is tailing. If the graph is like this, it is perfect. But if the graph is like this, it is fronting. Like this, it is tailing. So, while injecting the sample dissolved in mobile phase, the sample solution must be optimum in quantity, optimum in concentration. If it is highly dilute or if it is highly concentration, the results will not be perfect. So keep this thing very clear in your mind. Medium pressure liquid chromatography, column and its properties. Two materials are generally used in the MPLC column. They are silica gel and polypropylene as column materials. Columns are generally made of glass and plastic. It is so because to avoid corrosion and shield against damage. That is, the column is made up of glass, but it is covered with a plastic material so to protect it from breaking while it falls down or something dashes with it. It has varying length and a varying diameter. You are very much aware that when you vary the length, it can accommodate large sample. When you decrease the length, it can accommodate less quantity of sample. Varying the diameter, more would be the volume, and therefore more would be the sample. Smaller the diameter, less would be the volume, less would be the quantity of sample. So larger diameter and longer length is generally preferred when it comes to MPLs. The question is why? Separation or separating greater quantity of sample and with high resolution. The length is more, resolution will be good. The diameter is more, you will be able to load more quantity. That is the funda. That is the principle. That is the requirement behind MPLC. Because MPLC is a preparative chromatography method, and when it is a preparative chromatography method, you require two aspects, that my quantity of sample, which is then going to be separated into different required components, has to be in a very good quantity. So the quantity should be more. So to have more quantity, you should have a large diameter column. And even I want a very good resolution, a very good separation, very good efficiency. That is possible by increasing the length. So generally, the MPLC columns or the preparative chromatography columns are generally larger in length and larger in diameter. This is also a very good difference. What is the difference in the physical appearance of a column in analytical as well as preparative chromatography? In analytical chromatography, the internal diameter of the column is very much less and they are shorter in length, whereas in the preparative column chromatography, the internal diameter of the column is also large and their length are very much longer. This is the property of column that makes it applicable on the basis of preparative principle of chromatography. Medium pressure liquid chromatography involves adjusting the length and material size in the column. If the column length increases and you do not adjust the material size properly, your pressure will drop. I'll give you a very simple example. If I have a tube that is sucking water from one tank and throwing into the garden, if the length of the tube is 100 meter, it is throwing with a particular rate of 10 liter per minute. What happens to this rate if the length from 100 meter of the tube is increased to 300 meter? The pump will have to waste more energy, and so the rate of the water coming out will eventually decrease, or the thrust will eventually decrease. So it can be correlated over here that when you adjust the length, you also have to adjust the material size. If you forget to adjust the material size, the pressure will drop. This is something which was already discussed in basics in chromatography in the form of theoretical plates. N is equal to L by H. I can prove it over here. Length is increased, means L is more. If the H remains the same, number of plate is increased. Pressure might drop. But if you increase the size of each material plate, then again the pressure will be gained. So be very clear that adjustment of the length of the column, adjustment of the material size of the stationary phase filled in the column is very much essential so that the dropping of the pressure is restricted. Friends, some very basic topics, that what is stationary phase, what is mobile phase. I am not getting them in these videos because we already had an online lectures, online course basics in chromatography on that. Nevertheless, if few of you have missed those, the related video that are required are available in the description of this video. Along with them, at the end of this video, you will be able to get a direct link to those videos. So I request you to revise them if you have already seen. Visit those videos if you have not seen, because they are very much essential to continue in this series for better understanding. Medium pressure liquid chromatography has a fraction control collector. A fraction collector collects the fraction automatically I told you based on the lambda max of the detector. If it is a UV visible detector. The volume of the collected fraction depends upon the size of the column and the rate of flow. The more the size of the column because of the diameter, the more would be the quantity of the component separated and collected in the fraction collector. Fraction collector collects the fractions on a fixed rate and online monitoring of the separation peaks can be carried out. Fraction collector is only and only used when you are dealing with preparative chromatography because these collected fractions have to be used for further research. But if I am doing analytical chromatography, no need, just throw that out as a waste. Instrumentation part also has detector. The eluate can be monitored using thin layer chromatography of the fractions. Once the fractions are collected, go for TLC. Online monitoring can be afforded using single wavelength UV visible detectors. The detectors meant for analytical HPLC cannot be used to MPLC because of the loss in sensitivity due to high flow rates. Sometimes, due to high flow rate, the detector that are used or the UV visible detector that are used in HPLC are not the same that can be used in MPLC. You need few modifications. You need them to be more smart. You need them to be more sensible to detect the quantity of the sample. In order to compensate for the high flow rates and concentration, a splitter that splits the eluate from the column is used. Means the sample that is coming out of the column, the total quantity does not go into the detector. Very little quantity goes into the detector for detection and the rest remains out. So this is called a splitter. One of the split part is channeled to the fractional collector while the other fraction goes into the detector. I can show you over here in this figure. See, over here from the column when it comes out, it goes into the detector. Over here, it gets detects and goes towards the fractional collector. Over here, if I do not want to allow the complete sample to go into the detector, I can keep a splitter over here. It will allow a very less quantity to go into the detector and the rest will directly get collected into the fractional collector. It is a mechanism of the split mechanism or a split type of a channel that is used in the detector. A recorder is connected to the detector which renders the output of the separation in the form of peaks. So friends, these are some of the instrumentation part that is involved in MPLC, medium pressure liquid chromatography. Medium pressure liquid chromatography has applications in pharmaceutical industries, food industries, chemical industries, natural product isolation, and separating large quantity of biochemical compounds. So these are few of the applications of medium pressure liquid chromatography. So friends, this was an introductory part of the Advances in Chromatography online course through the first short lecture MPLC, medium pressure liquid chromatography. There will be two more lectures coming in the two consecutive videos that will be preparative HPLC followed by UPLC. Friends, this time based on the feedback from the basic course on online chromatography and even the feedback after the intermediate test, we have designed 12 to 14 lectures where these are the first three set of lectures, that is MPLC, preparative HPLC, UPLC. There will be no daily test, but after these three lectures, you will have a combined test on the day 4. On the 5th, 6th and 7th day, you will have LCMS, GCMS and one more combinational technique. The third week or the third set will be three more lectures on such three interesting research based topic, and again the fourth week will be a mathematical chromatography part. Pardon me, I'm speaking big, but the fourth set. What we have planned is three lectures, one test, three lectures, one test. So 12 lectures, four tests, and then there will be a final test for you. So friends, once again I thank you all for joining us in this online course, Advances in Chromatography. As officially it was the first lecture for Advances in Chromatography, we had kept it little short, but be prepared. Our lecture, according to our tradition, will be somewhat around 50 to 60 minutes from the next lecture. You are free to contact me. You are free to send me doubts. You are free to send me feedback, your queries, your suggestions. Keep supporting, keep learning, and be in touch. Thank you.